Hello. Hi. Yes, even the others are in class. You're actually cutting me short when I'm talking. Yeah, join the class. You're supposed to be in this group. Pardon? Yeah, it's online. Join the class. You use the credentials. Yes. So I have. Okay. So now, like I said, I will start with pre-lab 2 because there is sort of an overlap between these two lab sessions. So I'll start with what is related to nerves, and after I start with the nerves, then we'll go to what is related to muscles. So I had touched a bit in class about the compound action potential, but I have notes here to go with this pre-lab. So by the end of this activity, you should be able to describe and demonstrate the effects of an electrical stimuli of the nerves of the forearm. So you have to know where the nerves of the forearm are located, and when you stimulate them, what is the end result that you see. Number two, the most important thing that we are doing in this test is to check the nerve conduction velocity. Now, when you're looking at velocity, you're looking at the speed at which an impulse is transmitted. Different nerves will have different speeds, and we are going to look at specifically the median nerve. So you need to know a bit of your anatomy so that you know which nerve you're working with and where it is located in your forearm. Now, the other thing I need us to look at is we look at the Power Lab system, what it entails and what it is all about. So the Power Lab is nothing but a box, a small box, which is just the size of your computer, only slightly of a bigger width. And this Power Lab is the hardware unit. It's the one which has the ability to measure electrical signals, to take, to convert, to generate, to analyze. However, that box on its own cannot work. It needs to be fed with information, and the information is coming from you as the subject. So we must have some accessories which are going to connect the hardware, which is the Power Lab box, to you, the individual. So the accessories must be tailor-made for the particular experiment that they are going to carry out, and they must be able to connect to the hardware. Now, after they connect, like I said, the Power Lab can detect, can take and analyze, and even send information. But how are you going to read that unless you have a unit that can be able to show you in a screen or in a graphical format what the results are? So the Power Lab must also be connected to the monitor. And the monitor is just not like but a computer monitor. So once you connect those three parts, the accessories to the individual, the Power Lab box, and then finally we have the monitor. We need a software program that will be able to understand all this information that is coming to the Power Lab. It should be able to help in analysis. So this is the software that we work with, and it is known as the Lab Tutor. So what is the Power Lab in essence? This is a system that is used to record various physiological properties. It could either be for animals or for humans, depending on the kit that you purchase. Some of them are very tiny, so that the electrodes are tiny enough so that they can only fit in a very small animal, but others are large enough to measure the humans. So it is that whole system that will be able to collect data and do analysis, and you can actually manipulate the data, and then all this information is displayed in the monitor, and you can actually save it. So let me show you the real connection here. So I told you the Power Lab is nothing but a small box. This one, it is the hardware. In fact, when you go to the labs, I'm sure Hezron will introduce to you. Maybe you have worked with this is your first practical physiology class, but have you been shown the Power Lab using in another course, so that I'm not repeating myself?
No.
Oh, so this is the first time you're seeing it? Then in that case, I can introduce to you that Power Lab. It's a box, a small box. It looks like the decoder of your TV. So what it has the front side, or what you call the front panel here. And the front panel has actually been enlarged in this box here. So this is the front panel, and you can see that it has ports whereby you can actually insert cables. So this is giving you opportunity to insert several cables which will feed information to the Power Lab, or which will take information away from the Power Lab. Now, on the other side, behind the box, on the back panel, you can see if you turn this box around, this is what you are likely to see. It is fed, or rather it utilizes energy from the power, which is the power input here. But you can also print from this Power Lab, or you can also feed digital information to the monitor. So whatever information you'll be feeding to the Power Lab, say for instance, you want to give a few millivolts to the Power Lab, you are going to change the knobs here. So there is the on button, you change the knobs, which will tell you for each particular channel what you are feeding the Power Lab. Then on the other hand, there is information coming from the Power Lab. For instance, I'll give you a good example. If I want to introduce an electrical current, one of these jets is going to give the current to the power to you as the individual, but I have to have the accessory for me to punch, like it's a button, like here in this case, so that when I punch it, it will stimulate, it will send an electrical signal to the Power Lab. But remember, this button is supposed to be stimulating part of my body. So there must be a cable from the Power Lab to that part of the body, so that now it sends the current there. So there will be an input channel and an output channel. So as you can see, these two, they are output, and these four are input channels. They feed the information to the Power Lab, and these ones will feed information away from the Power Lab. Now, once you get that information, it will do the analysis. These buttons are just simply to change. If you want to increase the voltage, you want to decrease the voltage and all that, or even the timing. So these buttons will help you with that. However, on the back side, after if the information goes to the box, that information can be analyzed by the Power Lab, but then it has to be displayed somewhere. So there will be cables to connect the box to the monitor. So we have a power input cable, but then we have other cables that can connect to the monitor. Or if you want to completely go and store the information using a USB, you can do that. Or even if you want to print directly, you can connect it to a printer. So that is the main setup of the Power Lab. So now You are feeding it with information and it is analyzing and it is displaying. But the question is, what software does it use? So you have to connect it to the computer and feed the computer with a software that can understand the information that is coming, so that when the digital signal comes, it is analyzed by the computer and then it is drawn into a graph. And this is what you are going to see. You visualize, you observe the graph. Now the question is, once you observe, do you save it or do you print it or whatever you want to do with the data? So this will be the simple setup of a power lab. So one of the experiments you'll be doing is looking at the nerve conduction test. Now, this simple setup shows you, it actually analyzes what I have just told you here. We have an electrode here that can trigger information. So what will happen is, there is power within the power lab. If I turn it on, what it will do, it will send a current. And once it sends a current with one of these cables, it will reach here. So we have the input electrode and the output. So what will happen is that as it moves from here to here, it will connect through your nerve, so that it sends a signal and then the excess is picked up here and it is returned. So now, having completed that circuit, what it will have done essentially is stimulated the nerve that is located within your forearm. After it stimulates the nerve, the next thing is you want to record what is happening. Now you can see that here we have at the wrist, we have another cable that is attached, and this cable is supposed to be connected to the earth, so that if we have excess charges, we don't want to leave you charged, so that when you go and touch somebody, they start feeling a current of electricity passing through you. So what we do is, you must have an earth cable, so that the excess charges, if they don't go back to the gadget, then they are picked up by the earth and they are connected and they are dissipated where they should. Now, after you stimulate this nerve, you find that this nerve comes and innervates special muscles here of the thumb. So this, like I said, you need to understand what the nerves are. This is called the abductor pollicis brevis, and it is actually in one of the slides here. So these abductor muscles, what they do is they connect to the thumb here. And once this electrical current coming from the nerve reaches the muscle, you find that you have nerve terminal. When they reach the muscle, they are going to cause an action potential to run along the muscle cell membrane or the sarcolemma. So what will happen is, when this electrical current passes through the muscle, some of it will end up getting into one of the electrodes here, and they'll go and feed the power lab. And of course, you have to have an end here, a recording one. Here again, it will come, and we have one that is picking it up here. What are the changes? And then here, what are the other changes? So what it does, it takes a comparison of this charge to this charge, and we have a reading. And we'll see how that happens in one of the slides. So we have the first electrode and then the second electrode, and it will do a comparison of the electrical charges between these electrodes. So if you remember, you have the neuron here, and it has the neuroterminal, and this neuroterminal is terminating on a muscle. So now this will be the muscle. So it will generate a current here by releasing acetylcholine it will generate a current. And when it generates a current, there will be depolarization here. Now, what happens is that as these muscles, because it's not a single muscle, as these muscles are being depolarized, they will change the current here, and some of it will be picked by this electrode. So this electrode will tell you whether you had a positive charge or you have a negative charge because of this current or this depolarization. Similarly, we also have a second electrode here that will pick the current. So if the current is going in this direction, at one time I'll have a reading here, and then I will also have a reading here. And by comparing this reading to that reading, remember these are on the surface. They are surface electrodes. By comparing these two, I'll be able to tell of what electrical activity is occurring on the muscle. Now, I want to remind you here that you stimulated a nerve, and the reading you are getting is from a muscle. That means there was a connection between the nerve and the muscle. Yet you are reading electrical activity that is happening on a muscle. So you cannot dissociate neural activity from muscular activity. Usually you get them together. So I will show you how you do the nerve conduction test, but these are the basics of how you go and do, you analyze or you access electrical activity within the lab. So let us go to the basics of nerve physiology so that you can understand some of the things that I'm going to explain to you. So I have mentioned several times that you can have bundles of axons. Now, if you look at what I'm pointing at, this one, and you recall what I just taught you in the previous lecture in the morning, I told you you can have, this is a Schwann cell. From here to here is one Schwann cell. And from here to here is a Schwann cell that has been cut. Right in the middle, we have a yellow axon. So this is a myelinated neuron. And the axon is here, yellow in color, and you can see this is only one of them. So every cross-section of blue with a yellow center is a myelinated axon. On the other hand, you can see that you have a yellow fiber without any myelin, and this is an unmyelinated nerve or neuron. Sorry, this is a neuron, not nerve. But you can call it a nerve fiber because a nerve fiber is a neuron. So, by cutting a cross section, maybe I can have this one enlarged. By cutting a cross section, you're going to see the yellow ones are not myelinated, the blue ones have myelin sheath. In between, we have the red blood vessels and the blue blood vessels. The red ones have oxygenated blood, the blue are the oxygenated veins, so they are venues, and they are taking away the oxygenated blood. So all these are wrapped by a connective tissue. So we have all of them are wrapped together into a bundle with a connective tissue. Now, one of these bundles is represented by each of these cut sections. So in this case, we have one bundle here, we have another one here, we have another one here, another one, and another one. And all of them are wrapped together again into one bundle. So it's like a bundle within a bundle. And all of them are tied together into one nerve. So when you go to the lab, and you can see tweezers here, when you go to the lab and you use forceps, and you can actually lift up one white fiber, it is usually a nerve. And within the nerve, you have bundles, or what you call fascicles, and within each of these fascicles, there are other bundles of neurons. So this nerve is deeply embedded in your tissue. So around the nerve, we have muscles and we also have connective tissue. So for that reason, if I want to stimulate this nerve, I have to pass from your skin layer. I have to send an electrical signal from the skin, go to the muscle, and target the nerve that is deeply embedded within that area. And when I pass a current, that current will depolarize a given region, and only the neural fibers, only the nerve fibers that will reach threshold will be stimulated. The others cannot generate an action potential. So you have only few of them being depolarized enough to cause an action potential. So after you know the neurohistology, or is it physiology or histology, whereby you are just looking at the structure, even up to the microscopic structure, you also have to remember that once you start generating an action potential, it does not decrease in amplitude. So that action potential will be initiated at one point and it will go with the same amplitude, and you can go and detect it elsewhere. Now, as you can see, the nerves that you have are of different sizes. So the diameter can differ. It can be a small one. Compare this yellow one to this other small yellow one. So you find the diameter here is much larger than the other one. Now, when it comes to the speed of conduction, some of the things that will determine how fast they move will be the diameter of the neuron. So the larger diameter will have a faster conduction speed. Number two, if they are myelinated, they will even be faster than the non-myelinated neurons. So two of the things to take into consideration when you are looking at speed is the myelination and the diameter of the fiber. And they are the ones which will determine the velocity of conduction. Now, in most cases, the cell bodies, and I think I mentioned that in the morning, the cell bodies of the axons will either be within the CNS. For instance, if you have a motor neuron, the one that is innervating your skeletal muscle, you'll find that the cell body will be located within the CNS. On the other hand, if it is within, or sorry, it is outside the CNS, it will usually be located within a ganglion. So the cell bodies are not anywhere, just located haphazardly. They'll either be within the CNS or they'll be within a ganglion. So all these other nerve fibers that you find are usually the axons. So when you stimulate, you are stimulating at a certain part of the axon. Now, the other thing I had mentioned previously, for those people who missed the class last time, I said there are two ways of measuring electrical signals. So we can have what we call the intracellular recording, or we can have an extracellular recording. Now, if you have an axon, use a red color here. If I have the axon here, and this will be the cell membrane of the axon, I can look for a pipette, a small glass pipette that can penetrate right into the axoplasm. And then I have a metal rod here connecting. And what will happen is, if I connect this to a voltmeter and I put another electrode outside that cell membrane, what will happen is that ions are going to start moving from one side to the other. Why? Because at rest, we normally have the inside of the membrane is negative and the outside is positive. This creates a potential for movement of charges. So the minute you put a connection, it's like you are inserting a channel. You'll find that the ions will start to move, and as they move, they are going to be recorded. You'll find a pointer here is moving to show you the difference. Now, when they settle at rest, it will tell you that now that membrane has stabilized, and the point at which it has stabilized, the reading will be whatever value you're going to see here. So if it is for a neuron that is not stimulated, the reading settles at minus 70 millivolts. If you remember, I said in class that we do not deal, in the lab, we do not deal with voltage, we deal with millivolts. Otherwise, the volts are too much, they might cause electrocution. So now, when you have an electrode that is piercing the inside of a neuron, then in that case, the recording that you're going to see here is referred to as an intracellular recording. And it gives you the difference between the inside of a cell and the outside, or the inside of the axon and the outside. Now, this gives you the most accurate reading because it will give you a direct measurement. The problem is, given the diameter of the axons, it is very difficult to have this setup, especially with humans. The more advanced I would say the animals are, you find that the smaller the neurons they are, so that they modify the neurons to increase speed in different ways. However, with the invertebrate neuron, especially like the giant squid, this diameter is quite large, and this setup is possible within the lab. So there is a power lab kit, in fact we call it the animal kit, that will give you gadgets that are tiny enough to allow you to have such a setup, and you'll be able to read the membrane potential changes as you stimulate the axon. But that is only for the invertebrates. So now, when it comes to humans, our axon diameter are so small, this setup is a bit impossible. So what do we do? Then in that case, what we do is take a reading between two areas. So if this is the neuron, and I say that you have nerves, and the other thing is you don't want to have an invasive procedure. So you don't want to start poking things into the individual. So what you do is you place an electrode on the surface here. And this electrode can pick up a change in current. And then you place a second electrode here. And this one can also pick up a change in current. You connect these two electrodes via the voltmeter, and they will tell you, they will always be reading a difference between this electrode and the second electrode. So if there is an electric current passing here, it will be first picked by the first electrode, and then it will disappear, and then it will be picked by the second electrode. That way, you can actually even follow a graph and tell when it was picked here, when it was in the middle, and then when it was picked here and when it disappeared. So when you do this surface kind of recording, this is what we call the extracellular recording. Now, this is not as accurate as the intracellular because it is not telling us how many of these charges are moving. It is only telling us there is a difference between the first electrode and the second electrode. The advantage being that you can measure even the changes in these very, very tiny axons, and number two, it is a non-invasive procedure. So let us see what happens in these recordings. I've already shown you the setup here. So you find that as the electrons move, this pointer will move from one side to the other and it will stabilize at a given point. Now, with the extracellular, again, this is the difference. You'll be putting it on the surface. I think I've already explained to you what happens here. Yeah, this is what I wanted to see, the recording. So I'll go to the recording later. Now, with the action potential, this one just explains to you the setup. So I think you can read on your own. Now, what I want to look at is the recording. How is the recording when you have the intracellular setup versus the extracellular setup? When you have a lot of stuff, people tend to be distracted by the other things. So I will focus on this. You have the neuroterminal here and you have the axon. So when you look at this, you're going to read the action potential. And this is the action potential for a single neuron. So if you remember what we were doing when we were looking at the excitable cells, at one time, we have the resting membrane potential. And when we have that resting membrane potential, the arrows here is perpendicular. It doesn't move. Then when it reaches threshold here, you find the membrane polarity will rise, reach a peak. That means if it is moving to this side, it will reach its maximum and then it will go back. And when it goes back, it will hyperpolarize and then it will go back to rest. That is the normal shape of an action potential. So it all is in a negative format because all these values are negative. And then we have the positive format that goes and overshoots. And then there is that point of hyperpolarization here. So this is the kind of reading you are going to get. Now, this will be for a single neuron. Now, can you imagine when you depolarize and you have several neurons? So you have another axon here, and you have another axon here, several of them, and you depolarize. So this one will generate its own action potential, the depolarization and hyperpolarization. This one will also generate its own action potential, the depolarization, hyperpolarization, and the third one the same. So what are you going to be reading in this voltmeter? So because each of them is creating a depolarization, the charge that you are going to detect will be a summation of all these depolarizations. So this one, once it allows the sodium to get out, and this one allows sodium to get out, and the same here, when you check the membrane polarity at that time, it will be at that particular point a summation of all these small action potentials. For that reason, you cannot talk of an individual action potential. Instead, we are going to talk about a compound action potential. And what you're going to read is that as you depolarize and the depolarization is approaching that electrode, there will be a slowly increasing depolarization which reaches a point, a peak, and then some of it starts to go down, and then you find that it flattens out. So the shape is very similar to that one of a single action potential. However, this one is as a result of one action potential, a summation of two action potentials here, summation of several action potential, and here is when you have maximum number of neurons stimulated and all of them generating their own action potential so that you reach a peak. So once you start having the falling phase is because you have fewer and fewer and fewer neurons that are stimulated. So by the time you hit there, again, you may find that you have a negative phase. We talk of negative in that it is going below the polarization or the resting phase, and this is not attributed. Remember, hyperpolarization here is attributed to the slow closing potassium channels. In this case, that is not the case, usually because of a recording artifact. It has nothing to do with the channels. So the curve that you obtain here is as a result of summation of action potential. And this is what you are likely to get. So with the compound action potential, if I had placed my electrode here, so what will happen is that as the current passes, only the one with the minimum stimulation reaching threshold will be stimulated, so the action potential amplitude is just that level. But you find that as you increase the voltage, you stimulate more neurons, and each of these curves here is representing an individual action potential. So here you have more, and the summated curve is higher. Now, when you have quite a number of neurons, then you have a peak. But, however, you'll find that as the time proceeds, because when you're going in this direction, you're talking of time. Potential will determine the time. So that the first ones, the fastest to reach the threshold, you find that they will reach threshold, they will reach their peak action potential, and it will disappear. And then the next ones, which are next in line, the ones that have the next threshold is a stronger stimulation, they require more stimulus and they'll be more numerous. So with the compound action potential, this is the kind of trace that we expect. Now, I want you to look at these two recording electrodes. So if you remember, I told you when you have surface electrodes, we have both of them are placed on the surface of the nerve. So when there is a change in membrane potential here, it will compare this charge on the surface to this charge on the surface, and the pointer will move accordingly. But the reading that you are having is for a compound action potential. So it is a nerve with so many neurons and giving you a compound action potential. as opposed to this one, which gives you an individual. So with that in mind, let us look at this diagram or this animation, and we'll first cover that point. So imagine this gray part is wherever there is a gray part, look at the inside of the neuron. And the inside of the neuron has positive charges. So if I have my axon here, this is the cell membrane. So if I look at the inside, it's positive. Under normal circumstances, the outside is the one that is negative. So at this point where the reading electrodes are, they have a normal resting potential with the inside negative and the outside positive. So now it means that I have a depolarization here. At this point, I have an action potential. But you can see my electrode is not able to detect that because it is not immediately beneath the electrode. So as far as it is concerned, this is at rest because if you look at the charge of the reading electrode 1 and you compare it to the charge of reading electrode 2, they are the same. The inside is negative and the inside is negative here. Outside is positive, outside is positive. So what is the difference between this electrode and that electrode? And the difference is 0. When you have a 0 difference, then the pointer is perpendicular. Are we together up to that point? The pointer is perpendicular because the difference is 0. Are we okay?
Yes.
Yes.
Anybody who is lost, who doesn't understand that? Because I will go step by step, changing the charges. So the charge difference here is positive, but I cannot detect that. So my pointer is still at the negative position. Let's go to diagram 2. And you find that with diagram 2, these positive charges, remember it's an oxygen potential that is moving. Now the positive charges have reached where the reading electrode is able to detect. And what it is going to see is that the inside is positive as compared to electrode 2. So the reading electrode here is showing you the inside is positive as compared to electrode 2. So the pointer goes to the positive side. And if I were to draw a graph, when both of them are the same, the difference is 0. When the inside becomes positive, then that graph will go to the positive side. Remember, if I were to draw, what is it called? If I were to draw a graph, so that I have, this is 0, so this side will be positive and this side is negative. So as long as these two are the same, the difference is 0. So the line is straight, perpendicular. However, when this side becomes positive, the pointer shows you the graph goes up to the positive side. Now, if I were to put and compare the next graph, so for the next one, now look at both reading electrodes. Immediately beneath the electrode, both charges are positive. So what is the difference between electrode 1 and 2? What is the difference in charge between electrode 1 and 2?
0.
The difference is 0, which means if I was at rest here, and I was at 0, my graph goes back to 0. You see that? So now there is no difference between inside and outside. My pointer is perpendicular, showing me the inside is positive, the inside here is positive. There is no difference in charge between the two electrodes. So you have gone back to where you started. However, if you look at this graph, it is very different from this one. Because this one, the difference was 0, but the insides were both negative. Here the difference is 0, but the inside are both positive. And remember, if it is an action potential, it is still moving, and it is moving in that direction. Okay? It is actually going in this direction. So it will have to move. This positive block has to move and come to this side. So let us see what happens in the next graph. Now the next graph is showing you when this side, the positive charge, have gone to electrode 2. Now they will show you that electrode 1 is negative when it is compared to electrode 2. So if it is negative, remember here it was positive. Here electrode 1 was positive when compared to electrode 2. So when it was positive, it went up. Now that electrode 1 is negative, then it will go down. Can you see? It goes below 0. And now electrode 2 is the one that is positive on the inside. Let us see what happens when this one moves to the next block. So you see now the action potential has moved from this part to this part, but my voltmeter is showing me that the two electrodes have no difference in charge. They are back to resting membrane potential. So can you see the shape of the graph? It is at rest, it becomes positive, then it goes to negative, then it goes back where it was. This is a surface reading electrode, and this is the kind of reading that you expect when you place the two electrodes. If you remember, so let me just clear this and I take you back to this diagram. So remember, I told you, you place a reading electrode here and another reading electrode here. So that is the kind of reading you are going to see as electrical activity of the muscle, because it will be comparing electrode 1 to electrode 2. So when the current comes here, before it comes, both of them are flat. When it comes here, it goes positive. When it is in the middle, it goes down to 0. When it goes the other side, it goes negative. When it passes, it goes back to 0. That way you can tell the electrical activity of the muscle. Okay. Any questions so far? Yeah. So what are the characteristics of a compound action potential? So we have a latency period from the time you stimulate to the time you see the reading. So where you put, like now when I had told you, you put the stimulating electrode here. And this stimulating electrode, remember it is connected to the power lab. And the power lab, you have a button that you press to stimulate. So when you press in the power lab, it will show you that you have stimulated, and then it goes flat. Now below that graph, it will show you now the connection here between reading electrode 1 and reading electrode 2 going to the power lab. And so when you stimulate at that point, I'm going to get a reading at, say, this point. So I will have a positive and a negative, and then it goes back to 0. So now I have a time difference between the time I stimulated and the time I started observing the change in electrical activity. This period is known as the latency period. Okay? How long does it take for you to see the reading? Now, when it comes to, what is it called? When it comes to nerve conduction speed, you can place your stimulating electrode far from the target or near the target. So if you place it far from the target, you'll find that the latency period will be longer. So you find that this will be the latency period if you place the electrodes far. That is, the stimulating electrodes are far from the reading electrodes. If you place them close together, it means that the latency period will be shorter. Okay? So because of this, we can utilize this to look at the speed from this point to this point. And that is one of the experiments that you'll be doing. Because now, if you can be able to tell the difference between this point and this point where time is concerned, and you can measure the length between the first point and the second point. So you have distance, and number 2, you have time. So how do you calculate speed? How do you normally calculate speed or velocity? This is maths. How do you calculate speed?
Distance over time.
Distance over time.
Thank you. The distance here, you will use a ruler to measure the distance between, if you have the forearm here, this is your elbow, and this is your thumb, and this is your wrist. So at one time, we'll put the stimulating electrode close to the elbow. The next time, you'll put the stimulating electrode close to the wrist, and you will make a mark, and you'll be able to measure with a ruler. That will give you distance. Using the power lab, when you stimulate at this point, it will show you the latency period. If you stimulate at this point, it will show you the latency. And remember, the latency period is the time between you stimulate and the reaction in the thumb. Yeah. These are your reading electrodes. Yeah. So at one time, the first stimulating electrode will be close to the elbow, so you don't have the second one. And you'll measure how long it takes. Then you'll move this electrode from here and bring it here. And do the same, measure how long it takes. By deducting the time from here to here, you can calculate what is the time it takes to move this action potential from that point to that point. Now, if you have time, and using a ruler, you'll have measured distance. So now you can calculate distance over time, and it will tell you the speed of conduction. Okay? So that is one way you can utilize the latency period in calculating speed of conduction. Number 2, the other characteristic is that each of these compound action potentials has a threshold. So when you start working, we'll start with the minimum amount of voltage where we do not see a response. So as long as you look at the reading electrode and the line is flat, it shows you have not reached threshold. The minimum amount of voltage that you need to stimulate a response is the threshold value. So beyond that, the more voltage you apply, the higher the amplitude of the compound action potential. So you'll find that you have this kind of response depending on the voltage that you apply. So the minimum response will give you the threshold. Then the third one, we have the shape. Now the shape is very interesting because if you remember, we said that there is the positive and the negative and then back to zero. Positive, negative, back to zero. And what we'll find that the up shape or the down shape will depend on the reading electrodes, how they are placed, and that's why we say that it is the technique that will determine. It's the one that will tell us when the electrode goes below zero, if it is far from each other, what's the amplitude and all that. Now, when it comes to muscular activity, when it comes to muscle activity, you'll find that at one time you stimulate the muscle, it goes up and down. Another time, another set of muscles are being stimulated up or down. Another one up or down. So if you look at the activity of muscles, they are going, if there is a straight line, you can draw a straight line in the middle, but you find there is a lot of zigzag going up and down. So now, if you want to analyze muscular activity, it becomes very difficult. Why? If you add the negatives to the positives, you'll all get a straight line because the deflation or I would say the level at which it goes positive is equivalent to the level at which it goes negative. For that reason, if you add the two, you are likely to get zero. Now, to overcome this, what the Power Lab does, it takes the amplitude on the negative side and it squares it. And it takes the amplitude on the positive side and it squares it. Now, if you multiply negative times negative, what do you get?
Positive.
Yes, so whatever values you get here and you square them, they'll give you a positive value. Whatever values you get from here and you square them, they'll also give you a positive value. However, when you add the two, you find that now you'll have multiplied times 2, so you have to divide or get the square root so that you can actually get the positive deflation. For that reason, you find that depending on the activity, if the square here is very high and you draw a graph, you will find that this graph will show you the level of activity. So in this case, if this is 0, you find that the activity is all positive, and when you start decreasing, the activity is still positive, but you get a straight line. For that reason, you can be able to compare activity at this point and compare it to activity at that point. So all this will be shown in the Power Lab, just because of that simple calculation of using the square root and adding, or rather the squares and getting the square root to get this value. So that shows you the nerve conduction tests. And I think this just simply shows you the setup. Compound action potential, here is a stimulator at the elbow. You stimulate at the elbow and you read here and you get your reading. On the other hand, this is the elbow here. You stimulate, get the reading. That is the latency period 1. You remember when I told you you can have a long latency period? Then when you bring the electrodes close to their target muscle, they have a shorter latency. So you are going to take this latency period, deduct this one so that you can get the time and the length component. You use a ruler to measure. So you are going to do that experiment in the lab. Now, having seen this, it will be much easier. In fact, I won't go through all the slides in the second one. I will just show you 2 slides which will show you the difference between EMG what I have shown you. So with that kind of setup, you see that this one is actually a repeat of what I was showing you. The only difference is it's showing you how you involve the brain. The brain sends a signal through motor neurons, and these motor neurons are going to send a signal to the muscle. So there are 2 things that you can do in this setup when you're looking at the basic physiology of sensory and motor neurons. So what you can do here is you can put electrodes here and stimulate and observe the changes in the muscle. Alternatively, you can use your motor cortex, because the motor cortex can send information and stimulate muscles. So either of those 2 ways, you can stimulate muscular contraction. Now, with that in mind, I will skip this one because I can see I'm short of time. But you see that all these will be telling you the same thing. EMG, still similar to what I have told you. I think I'll do this in details in class. What I want to do is to show you the setup for the other experiment. So in the second experiment, we'll be using muscular contraction. So you can either recruit 1 motor unit or you can recruit more motor units, depending So this is the same setup like I had shown you. So let me go straight to this diagram. So for instance, here you have the biceps and the triceps. So when I ask you to fold your arm, we are going to contract the biceps. Now imagine if I'm trying to push your arm in this direction, and I'm still informing you to contract the muscles. What will happen is that you'll be sending more firing inputs to those muscles, trying to keep them so that they can overcome that resistance. Because of that, if I have reading electrodes here, they will show me an increase in muscular electrical activity. Just because it may not have moved, the arm may not have moved, but this arm, the distance may not have moved, but the fact that I'm applying resistance, you are firing more, there is a lot of electrical activity which can be detected by the motor neuron. Now, there are 2 experiments that you are going to do. In the first one, you'll be contracting the biceps and checking the kind of electrical activities they are. you are going to offer resistance to those biceps by placing weights on your arm. And this weight will be slowly increasing. And as you increase the weight, we are going to check what is the change in electrical activity. The second experiment, we are going to see what happens to the antagonistic muscle. So when you are contracting these muscles, what is actually happening to the antagonistic muscles? In most cases, people would respond that they relax. This will contract while this relaxes. But instead, we are going to see that when you do that, I have this graph. This graph, the top graph is showing you the activity of the biceps. So when I tell you to contract slightly, this is the activity. Now look at what happens. At the exact same time you are contracting the biceps, look at what is happening to the activity of the triceps. At the same period of time, there is an increase in activity. When I tell you to contract the triceps, this is the increase in activity of the triceps. Look at what happens in the biceps. So this phenomena where both antagonistic muscles are being stimulated is known as the core activation. And given that it is time, I might as well I think I will allow you to go to your next class. I will carry this in details when we come to the class session. But at least the two concepts I've given you is when you offer resistance, you'll do that, and you'll see the change in electrical activity. Number two, when you change the weights, you'll see what happens to electrical activity. And number three, when you do the experiment of core activation. And we'll see the significance of this when we go to class. So far, any questions? Any questions? How many are present?
What time is the experiment on Monday?
You are scheduled to start at 9 o'clock.
Which lab?
All right.
Pardon?
Which lab?
It's in lab D. It's the physiology lab at the corner.
All right, thank you.
Okay, so quickly, I will still take attendance for this. Nawal, are you there?
Yes, I'm there.
Sumaya? Sumaya is not there?
Present, madam.
Okay. So quickly.
Ma'am.
Okay. And Dorothy Kajuju?
Present.
Miss Gila is present.
Gilo? Yeah, I've seen her.